right. Good afternoon, everyone. My name is Gio. My virtual virus presentation will be on the cardiomegavirus, abbreviated CMV, CMV1. I will be referring to the virus as CMV1 throughout the entire presentation. So let's begin. All right. So here we have the CMV1's viral structure. CMV1 is an enveloped double-stranded DNA virus that falls under Class 1 Baltimore classification system. We can also see that it has a non-segmented genome, and the DNA will begin to circulate once inside the host nucleus. We'll get into that a little bit further into the presentation. As you can see on the screen, we have the GP and GM glycoproteins, GP depicted in yellow, which will be the primary attachment glycoprotein, and GM in black, which assist in membrane fusion and entry. We can also see that the virus is about 150 nanometers in size and contains an isosahedral capsid, which surrounds the double-stranded DNA genome. And moving on, I have the little origin story, if you will, of the CMV1 virus. CMV1 was originally created by biomedical engineers as a synthetic therapeutic virus intended to strengthen cardiac tissues under stress. However, mutation in its GP glycoprotein, as we saw earlier, is now able to recognize Nectin-1 within the within the respiratory tract, which led to unintended human infection, little outbreak of our CMV1 virus. And moving on to the next slide, I kind of broke the CMV virus into two phases, if you will. In this slide, I'm going to be going over its respiratory phase, and then as we go on to the presentation, we'll dive further into its replication. All right, so let's begin with the respiratory phase. The infection will begin, like I said, in the respiratory tract. The glycoprotein will bind to now the Nectin-1 receptor in respiratory epithelial cells. Once this occurs, the virus will enter through receptor-mediated endocytosis, and we'll also see some help with the GM glycoprotein to help promote envelope fusion. Once the envelope fuses with the membrane, the capsid and the tegument are now released into the cytoplasm and then transported into the nucleus. Once inside the nucleus, we're going to see some immediate early genes begin to be transcribed by the host RNA polymerase 2, as well as some early gene replication. However, this is more just a limited replication, just enough to produce a small number of virons for shedding to be further let into the bloodstream. Moving on, this was the diagram I was referring to a little bit ago. I will just, go over both points, and then the next slide will highlight the important things that I mentioned throughout this portion of the presentation. So as I mentioned before, the life cycle began in the respiratory tract, where the GP glycoprotein will bind to Nectin-1. And as I said earlier as well, the receptor-mediated endocytosis will occur, and then the capsid is then released into the cytoplasm and is transported to the nucleus. Inside the respiratory epithelial cells, the virus will perform, like I said, low-level replication, just to get these early genes expressed and to produce that small number of virons. This phase allows for shedding through droplets, which also creates enough progeny to enter the bloodstream to produce viremia, which again will expose that virus into the bloodstream to then travel to areas such as the heart, where CMV1 will begin to do its most damage, air quotes. But so once in circulation, the virus will begin to travel to the heart, where it will encounter its needed receptor, the calyx 3 beta receptor on cardiomyocytes. So this binding to this receptor will initiate its second, more productive phase of the life cycle. Now, once inside the cardiomyocytes, the virus will begin a full lytic replication cycle. Here, it's going to be the same rinse, wash, and repeat process. We're going to see some immediate early genes begin to activate host RNA polymerase, and early genes will produce the replication machinery, and the viral genome will enter its rolling cycle replication. And then moving on, we're going to see obviously some late gene expression, which is going to produce those structural proteins, the capsid, the tegumen, and the envelope glycoproteins, which are then assembled into mature virons. These virons will then acquire their final envelope in the Golgi and exit through exocytosis. And as replication intensifies, we're going to eventually see the infected cardiomyocyte undergo cell lysis, which then will release a large number of virons into the surrounding cardiac tissue. This lytic release and spread between these neighboring heart cells is what's going to cause viral myocarditis, and which will drive the major pathology of CMV1, which we will discuss later. Sorry. And these are just the main points I brought up throughout the diagram. We're going to see that systematic spread, penetration and fusion, the uncoating of the capsid at the nuclear pore, thus releasing that double-stranded DNA genome into the nucleus. And just, again, that brief overview of its propagation, where, again, it's going to follow that lytic propagation strategy. We're going to see that binding. Once that binding to the calyx 3 beta receptor is initiated, the virus will begin to replicate aggressively, thus rupturing those cells and then contributing to that viral myocarditis that we had mentioned earlier. And then moving on, we have the host interaction, and just, highlighting the heavy points that CMV1 will rely on the host's own machinery. So as I mentioned before, the host CMV1 will use the host RNA polymerase to transcribe its viral mRNA. Those transcripts will receive a 5' cap and a 3' poly A tail using the host capping and polydylation enzymes, just like normal host mRNA. We'll also see that CMV1 will hijack host ribosomes to translate all viral proteins, as we saw in the diagram here. The ribosome is translating all those viral proteins. And we will go, we also see that the host nucleus, use the host nucleus for DNA replication, and then it will redirect the ER and Golgi apparatus to fold and assemble those viral glycoproteins. The tegumen proteins from CMV1 will also block the interferon signaling, which will allow the virus to replicate with less pressure from the immune system. And as replication furthers and gets more and more aggressive, we're going to see that cardiomyocyte metabolism will become overwhelmed, thus contributing to tissue damage in the body. Which brings me to the pathology. I mentioned a lot throughout this presentation that the infection of the heart is going to lead to that myocarditis, which is that just inflammation of cardiac muscle tissue, which you can see in the picture depicted. For this normal heart, we're going to see, just normal myocardium, the heart muscles. However, you can see in the picture on the right, the myocarditis, you can see just a thick and inflamed, swollen heart muscle tissue. And that is because the virus, again, uses that lytic replication strategy within the cardiomyocytes, which will the infected cells will eventually burst, not in the heart itself, but within the tissue around the heart, causing tissue injury, inflammation, as well as, rapid fatigue and other palpitations that we can see for the symptoms that will happen with CMV1. Oh, and things like oxygen deprivation will definitely be a factor in this as well. So those who are at risk, this isn't the virus that is not the virus that they do not want to contract. And yeah, here are my references, and that is the end of my presentation. I just want to say thank you guys so much for your patience in watching the presentation. I hope you all have a great Thanksgiving. I wish I was there. Thank you.